Hi, in this video we'll continue our discussion about methods used to engineer genomes in DNA. The first thing we'll talk about is ligating two pieces of DNA together, such as when you're putting a sequence into a plasmid. We'll talk about transformation, that's putting that stitched together plasmid into a new organism. We'll talk about how we can make a genomic library in which we'll store every piece of a genome in small parts. We'll talk about how to identify specific clones in such a library, picking out maybe the gene you're interested in. And we'll end by talking about the important step of taking mRNA and converting it into cDNA, which is a useful way to manipulate human genes because now the cDNA, since we've made it from the mRNA, no longer contains the introns. It's usually just the coding part of the gene with some regulatory sequences on the ends. So we saw in the previous video how we were able to cut a piece of DNA into two pieces using restriction enzymes, and here we're going to put them back together. By far the most commonly used method is the joining of two complementary staggered ends. Here we have two pieces of DNA that were cut with a restriction enzyme that leaves a TTAA overhang. These will base pair with each other, the A's and the T's and the A's and the T's, and then we'll stitch that together with ligase and ATP. We'll make a bond between the 5' and 3' ends of the sugars. You can also join two blunt ends, meaning there's no overhang. This has the advantage of you being able to put any two pieces together without the restriction that they have complementary bases. The disadvantage of this is that it is much less efficient in terms of putting pieces together again. The overhangs really assist that process. Lastly, how do you join non-complementary ends is, well, one thing it's called you have to polish the ends. In this case, we wanted to join a staggered piece of DNA with a blunt piece of DNA. And the way that you do that is by a combination of chewing back the overhang and filling in the exposed bases. In this case, the diagram shows that you're filling in the bases, and now you've got a blunted end molecule. You're going to combine it with this other molecule, and you can see that we've stitched that piece together right there. Because of the extra steps involved, you only use this method when you absolutely need to. And this is just showing a little bit of a detail of the step that occurs. Again, here we have two bases A and G. There's a phosphate group and a hydroxyl group, and what we're going to do is we're going to form the back bond with the phosphodiester linkage between the two bases. In this process, we're going to take advantage of the ability that cells have to take up DNA. In this case, we have one bacterial cell which is dying. In the process of dying, it lyses its DNA. There's a neighbor cell which identifies a piece of DNA and will take it up, and then it may or may not incorporate into the genome. Most of the time it doesn't, but occasionally it will, and in that case you have transferred a gene from one bacteria to another. So we're going to talk about making a specific piece of DNA, and in this case we have a plasmid or a vector, a generic vector, and we have a piece of DNA that we want to clone or propagate inside bacteria. Now, a cloning plasmid has to have three things. It has to have a multiple cloning site, which is a number of restriction enzymes that can be used to cut the plasmid open and insert the piece of DNA that you want to insert it. Second thing is it has to have an origin of replication, which I've indicated by this blue dot, and it has to have an antibiotic resistance gene, or more generically referred to as a selectable marker, and that I've indicated as a green dot. For the case of the bacteria, it is almost always an antibiotic resistance gene, and this is something that the organisms are going to need in order to survive our process. So we will start by cutting both the plasmid and our piece of data that we want to insert. This is probably from a PCR reaction, so it's got blunt ends, but at the ends they have a recognition sequence. We're going to cut the plasmid, we're going to cut the PCR product, they're going to leave these TTAA overhangs, which are going to be complementary. This overhang will bind with this overhang and create a new molecule, which is held together lightly by these 4 base pair overhangs, and then we'll use the enzyme ligase to form covalent bonds between the backbone, and now we've got a single DNA molecule, double-stranded DNA molecule, which is a plasmid with the insert of our interest, and then we're going to put it into bacteria. So here we have our cloned plasmid. We've got the insert sequence, the origin of replication, the antibiotic resistance gene. We're going to insert this plasmid into a bacteria, and that will usually be done with the aid of an electric pulse or with some lipid reagents that help make cells a little bit more permeable. Once inside a cell, you've got the plasmid, and you also have the bacterial genome, which is not drawn here. We're going to put this inside an Erlenmeyer flask. We're going to grow this up. Now, inside the liquid media here, we're going to place an antibiotic, which kills bacteria. The only bacteria that will survive this is the bacteria that has the antibiotic resistance gene. So cells without this are going to die, and that's important because at the end you want to have most or all of the bacteria having the plasmid that you're interested in. Once these bacteria grow up, then you can isolate the bacteria, you can lyse them en masse, and isolate many millions and billions of copies of the plasmid molecule, which you can now do various things with. And one of the things that you might most like to do now that you've cloned a sequence and put it inside a bacteria is to use that sequence that you've inserted to make a protein which might have some utility for you, either scientific or commercial. You've engineered a bacteria to become a factory for the protein of interest by inserting that gene into an expression vector, another term for the plasmid. Now, one particular type of cloning process that people have used both for gene discovery and genome sequencing is making something called a genomic library. And here we would take human DNA, we would cut it with a restriction enzyme into many billions of pieces. We can take all those pieces in one tube, mix it with a lot of copies of the plasmid. Since they've all been cut with the same restriction nuclease, they all have the same overhangs, they're all going to get inserted into a plasmid sequence. We can take this complex mixture of plasmids, put it into a large number of bacteria. In nearly all cases, only one plasmid will go into individual bacteria. If 2 go into a bacteria, they will frequently lose one of them because you have redundant antibiotic resistance genes. But now this collection of bacteria is referred to as a genomic library because in principle an entire genome is represented in small pieces inside bacteria. Now, if you're interested in a specific clone or a specific gene that's present in one of those bacteria of a genomic library, you will grow many of those bacteria up on a petri dish. Again, you'll take a piece of plastic, you'll put it lightly on top of the plate. Now you will, on this top of this piece of plastic, you will lyse the cells, but the DNA, which is negatively charged, will stick to the plastic. And now you can do a hybridization experiment where you have a label that is going to bind to some of the DNA, and you'll identify which are the locations with a piece of photographic film to see where the radioactivity occurred. You'll see which of the colonies are those of interest, and I believe, and it can be tricky in the lab, that it's this colony and this colony, at least that looks like it to me. And then you would take with a toothpick a small bit of that bacteria, place it in a test tube with some liquid media, grow that up, and now you have a bacteria which has a specific gene from your genome cloned into it. Now, in the last few years, a new method has been developed which does not require restriction enzymes or the handling of individual pieces of DNA one at a time, and this method is called Gibson annealing or Gibson assembly, and Wilson Wong told us a little bit about this. But to stitch two pieces of DNA together, all you have to do now is have complementary sequences, maybe about 15 base pairs that are the same on two pieces of DNA, and you can usually put them there very easily by PCR. Now what you're going to do, you're going to put them into a tube. You're going to have a 5' exonuclease, which will chew back one of the strands of each of those two. It's only going to be able to chew for a small amount of time because you're going to make it happen quickly. Now the two sequences of the chewed back portions of DNA are complementary. They will overlap. They will form a combined piece of DNA, and now your polymerase will come in and it will extend the fragments to fill in the gaps, and then ligase will seal the nicks, and you've put together two pieces without ever using a restriction enzyme to cut them there. All that you've been required to do is have overlapping bases on the ends of DNA. And as this example shows, that you can stitch many pieces together in a single step, because the only way that these are going to eventually produce a complete piece of DNA is the overlap of this promoter region, for instance, promoter PCR product, which overlaps the plasmid, and then it has these different elements: blue, purple, pink, orange, and then green with the vector again, orange here and green here. And so the only way you can make a complete piece of DNA is if all these things overlap, and this can happen simultaneously, which is opening up new possibilities for both longer pieces of DNA being stitched together and more complex pieces of DNA being assembled rapidly inside a laboratory. Now in the lab portion of the course, you're doing something very similar. You have a plasmid with an antibiotic resistance gene, a multiple cloning site, origin of replication not shown. You're going to use two restriction enzymes to cut open, and you have a promoter fragment, which is PCR'd, which you will cut with the two restriction enzymes. And you do this, you use two restriction enzymes in this case so that you can specify the orientation that this is going into the plasmid. You're going to ligate the plasmid. You're going to transform it in lab 6, put it into bacteria. Here you can see the bacterial chromosome and your plasmid. You're going to put this either on a plate or into liquid media, and only those cells which have the antibiotic resistance gene are going to grow up. And then you're going to analyze the expression levels of a protein. Now, different people in the lab are going to be putting in different promoter fragments. Some promoters are going to be strong. It's going to produce a lot of protein. Some promoters are going to be weak. They'll produce smaller amounts of protein. And the one other thing that you'll do is you'll do a quality control step where you're going to use PCR, which we'll see in the next video, to ensure that the proper piece of DNA that you think this promoter thing has gone in this plasmid. And you'll do that by doing a PCR with one primer on the plasmid and one primer on the insert. So again, you'll be PCRing across a junction, and that will verify that you have the right piece of the right construct. The last thing to discuss, which is essential, is making cDNA, which is complementary DNA, from messenger RNA. Again, messenger RNA is nice because all the introns are removed, and we also always have a poly A tail. Remember, that's one thing that eukaryotes do. So that allows us to take a poly T primer, which will hybridize with the poly A tail, and now we can synthesize a strand of DNA from the original mRNA molecule. mRNA works perfectly fine as a template. Then we're going to add an enzyme, in this case RNase-H, which will cleave the RNA of an RNA that's in an RNA-DNA hybrid, and then we'll be able to use these RNA fragments as primers, which will allow us to synthesize a second strand of DNA. And now we've got double-stranded DNA, which we always will indicate with the C there to let you know that this is DNA made from mRNA and containing a gene without introns. Sometimes people will specify gDNA to indicate genomic DNA if you want to clone one of those fragments. And now that you've got a double-stranded copy of a gene, you might clone that endoplasmid to make the protein that you want to produce, as we showed in the previous example. Okay, thank you for listening.